Hello, everyone, and welcome to today's webinar, Unlocking the Power of SH800 Cell Sorter, Insights from Gene Editing to Immunology. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational webinar is presented by LabRoots and brought to you by Sony Biotechnology. To learn more, visit sonybiotechnology.com. We do encourage you to participate today by submitting your questions during the presentation. To do so, simply type them into the Ask a Question box and click Send. After the presentation, we'll try to answer as many as we have time for. If you do have any technical issues, you can also use that Q&A box and we'll assist you directly. Before we get into the presentation, though, we do have a couple poll questions we'd like to ask. The first one here is, what is your area of research? We've got CRISPR, immunology, stem cell biology, cancer research, genomics, and other. Give everybody a few moments here to answer those before we move on to the next question. Thank you for answering that. We'll just give it another few seconds here before we move on to the next one. The next question here is, where are the cell sorters you use located? In our research lab or department, in the core share flow cytometry facility, in a satellite lab located in our institute, or I do not use a cell sorter at this time. Give everybody, like I said, a minute or so to read and answer the question. See the answers coming in. I appreciate that. And finally, would you like to learn more about the Sony cell sorter and its capabilities that will be discussed in this webinar? Two yes options there and a no. Like I said, we'll give everybody time to answer this, and then we will move on to the presentation. All right. I'd like to introduce our speaker, Andy Draghi, Field Application Scientist at Sony Biotechnology. And Andy, you can go ahead and begin your presentation.
Hello, and thank you for joining me today. My name is Andy Draghi, and I'm a Field Application Scientist with Sony Biotechnology. During today's seminar, please hold your questions till after the talk concludes, as we have a Q&A at the end of the session. With that, I'd like to begin my presentation, Unlocking the Power of the SH800 Cell Sorter, Insights from Gene Editing to Immunology. As you can see from the image of the system, the SH800 is a benchtop fit plug-and-play system. When looking at the dimensions, you can see that this system can fit right on top of a table with a fluidics cart underneath. The SH800 is a microfluidics-based chip sorting system. It has advanced automation for setup, acquisition, sorting, and analysis. It has intuitive software that's going to support sorting into both tubes and plates, 96 and 384 well. Also available is a custom-designed biological safety cabinet if you desire that for your workflow. A little bit more on optics design. The system is available with up to 4 lasers, the 488, 405, 561, and 638 nanometer laser. There are multiple cameras and sensors to support automation. Daily alignment of the lasers to the chip ensures consistent results from day to day. We have a series of images that show a calibration image that the instrument uses to calibrate the stream, the droplets, the break-off. We also have a droplet image that the user sees that allows them to visibly appreciate their droplets. We also have a side stream image down at the bottom that shows the side streams that help, again, with the automatic calibration. On the right-hand side, we have a figure that describes the laser module, again, with up to 4 lasers. We then have the detection module, again, which is flexible in terms of filter application. You can swap out the filters based on your needs. And then if you look through the optics design schematic, you can see we have numerous cameras that support the laser calibration, the droplet calibration, the delay, the break-off, the side streams. They give you perfect calibration every time, all the time. A little more into the optics design. You can choose to have your system with up to 1 or maybe 4 lasers, depending on your needs. And again, this can support 6 colors. All 4 lasers are combined into a single laser beam spot. The PMT layout for the optical filters is user exchangeable. That means if you have the need for close neighbor fluorochromes, such as GFP and YFP, you can change out the filters and use that in your workflow. Automated daily alignment, again, is going to ensure you're going to have a robust, consistent signal from day to day. Moving on to fluidics. The sorting chip designed for use in a wide range of cell sizes. The system can run a variety of cell or chip nozzle sizes. So we have a 70, 100, and 130 micron chip. Each chip is individually wrapped, it's sterile, and it's serialized with a QR code. Upon scanning the QR code, baseline settings specific for that nozzle size are set up in the instrument automatically with no user input. Chips can be run for up to 24 hours and can be used multiple times within that 24-hour period. Here we have the basic layout of the chip. Vacuum line port, sheath line port, sample port, laser intersection or detection window, and then at the very bottom is the nozzle that forms our droplets. A little more on the fluidics based on our various sorting chips. Again, we have 3 sizes, 70, 100, and 130 micron. Fastest sorting is with that of the 70 micron. Used for the smallest cells, so under 10 microns in diameter. Bacteria, yeast, RBCs, splenocytes are ideally run on this. Our next size is our 100 micron chip. This is the most common chip size. Again, most cells are slightly smaller than 20 microns in size. PBMCs, K562, macrophages, stem cells, bone marrow are often run on this cell size or this chip size. The 130 micron chip is best for viability for those large, fragile cells, tumor preparations, disassociated tissue. It's going to be our slowest sorting chip. A lot of times people will use, fibroblasts, neurons, large irregular morphological cells, hybridoma, embryonic stem cells, and so forth. Again, choose the chip that's best for your workflow. This system offers flexibility. A little bit more about microfluidics chips, the benefits. Again, it's aligned daily for optimal optical performance. This system can easily recover from clogs or air bubbles using software options. Chips can be exchanged to eliminate the concern for a sample carryover. If we want to deal with mitigate clogs, again, we can have a clog that maybe forms here and occludes flow. So flow stops. We do a back flush, removing the clog, and sample flow resumes, and we're back to sorting. Again, with a push of a few buttons with the software, we're back up and running. Moving on to the SH800 cell sorter key benefits. Again, automated setup, a lot of automated features with this system. System priming, chip alignment, optimized break-off, side stream adjustment, and droplet delay measurement is all done automatically. Add some beads, walk away. The system will get itself up and running for your daily sorts. Here we have an image that shows chip insertion. And then upon successful completion of the automatic calibration, here is our window that displays that success. Now moving on to the software to kind of describe a basic workflow. Again, the system's really intuitive. It has software to help you create your experiment, build your plots and your statistics you need for your sorts. There's also a compensation wizard that you can use in conjunction, with the software that'll help you, create a matrix to deal with any compensation concerns that you may have. Again, no prior knowledge. And flow cytometry is really needed because the wizard is so efficient at walking you through all the required steps. Once you got your compensation done, you can move into acquisition and acquire some data, confirm your gates, and set up your sorts, all with easily guided workflows. Now that we've moved into sorting, how can you sort? What are your options? Well, you can bulk sort, two-way sorting into tubes or conicals. Again, it could be Eppendorf tubes. It could even be cultures tubes if you're doing yeast. And you can also sort into plates, 96 well, 384 well plates, giving you flexibility in how you capture your cells for your downstream applications. Just to walk you through the internals of the sort chamber. Again, here we have our multi-well or plate holder that's angled. Here we have our tube holder. Right here are our deflection plates. Waste collection. Collection tubes are located here. The tube holder and the collection stage. How you sort your samples. You can bring your samples in a variety of ways and means. It could be an Eppendorf tube, could be a 0.5 ml, could be a 1.5 ml tube, or even a 2 ml tube. Of course, you can use the standard 5 ml fax tubes or the 15 ml conical tube. Tube holders can be agitated to keep your cells suspended. And above all, they can be cooled, so maintaining viability. No sense sorting cells if they're going to die. Sorting tube holders, so you can collect into a variety of tubes as well. Those could be your, standard 15 ml conical, 5 ml fax tubes. Again, you can also set this up to, sort into an Eppendorf tube if you want to, or even, as I witnessed the last couple of weeks, people will sort into, yeast culture tubes directly in the tube holder. Again, collection can be cooled. Plate holders are designed for a variety of plates, so no specific plate is needed. They could be round bottom, they could be flat bottom. Again, 96 well, 384 well. PCR plates, again, 96 well, 384 well. You could place this in the refrigerator ahead of time so that when you load your plate with reagents, it maintains the stability of those reagents. We even have an angled plate holder at 7 degrees to ease your setup and deposition of cells to the center of the well. One of the amazing features of this system is you can get indexed analysis with the SH800. What is index analysis? Well, index analysis records the XY coordinates of each sorted event. So if you want to have a certain cell, placed in a certain well, you can track the phenotype of individual sorter cells, in each well. Annotated cell data, single cell genomics, and cell cloning. This is critical information. So here in this example, we have a well. If we click on this well or a group of wells, we can actually see where each of these events lays out on the plots that we set up during our sort. A little more in index analysis, the well select mode. In this example, We've sorted using linear gates on a histogram. We have a variety of intensities of signal. And again, if we click on a well and we look as to where those cells came from, we can see, based on signal intensity and maybe SSC or complexity, where those events, landed. So we see the whole progress of that event from forward side scatter, the singlet gate, all the way to our definitive sorting gate here. Again, a very powerful feature that's critical in interpreting your results and your downstream application with your sorted cells. And to kind of, go over this a little bit more, again, there's a variety of ways you can use this, whether it's gate, single well. This information you'll soon realize is probably the most critical part in, analyzing your application. You had success with a certain marker, you had success with a certain expression profile. Why was it successful? Well, maybe it had higher GFP, maybe it had lower GFP. Maybe where it landed in one of your plots is actually what's dictating, downstream outcomes or phenotype differentiation. This information is critical for the sorting of your cells, and you can get this with the SH800. Let's move into research applications. The first one we're going to talk about is CRISPR-Cas9 screening. So, as many of you may know, millions of cells are transfected or transduced with GFP tag, Cas9, or maybe some other tag system. Cells are then sorted based on GFP expression. The sorted cells are screened for GFP expression, and clones are screened by transcriptomic profiling to identify those clones bearing, a desired mutation. So again, not a complex workflow, but very critical to identify these GFP positive cells, and then taking that data and interpreting the transcriptomic profiling data. A little bit more about CRISPR-Cas9. A lab that is successful with the SH800, hails from the University of California, Berkeley, the Savage Lab. And again, they're engineering Cas9 proteins and fluorescent biosensors. The benefits they feel are that they can have multiple users up and running and fully trained on this system quite easily. Even non-flow experts are able to gain access to this instrument at off hours. It's their personal sorter. And because they're able to get, round-the-clock access, weekend access, they can develop, optimize, their CRISPR-Cas9 cell libraries, their workflows. It's an iterative process to optimize these assays. So having, unrestricted access is critical to getting the workflow and productivity. A quote from Dave Savage, the SH800 opened the door. Complicated, time-intensive experiments could be done easily, allowing the lab to create a platform of technologies engineering different types of metabolite biosensors. And again, none of this is really, complicated or challenging. It's pretty much a straightforward sort, using a 70 micron chip, GFP tagged bacterial cells, from the library are sorted with CRISPR-Cas9 expression. The run rate is, quite impressive with 30,000 events per second to allow for a 40-fold purification. So again, this is our pre-sort data right up here, and we're looking to enrich this population. And here's our post-sort data here. We're basically looking at one color, but again, a marked increase in purification, allowing for downstream applications to be successful. As with a lot of sorting projects or assays, a lot of times there's multiple sort steps, such as the case of the CRISPR-Cas9. Successive rounds of sorting for the protein of interest is conducted. So in this example here, we're sorting GFP positive cells after induction. You may or may not wish to actually look at the the GFP negative cells that don't have induction, depending on what your needs are. But then, you might have to go through another round of GFP positive cell sorts, looking at the induction to isolate those active switches. Moving on to the benefits of the SH800 for CRISPR applications. Again, none of these sorts or assays are really complicated. They're not high parameter. But at the end of the day, if you couldn't put this in your laboratory and have it run reliably and consistently, that would be a problem. If you couldn't have people easily use the system with automated features and calibration and walk-away features, it might be more challenging for your lab to actually adopt this. Well, the SH800 is highly automated, calibrates itself. There's walk-away operation. There's wizards to help you do key features and critical steps. Capability. The system is able to run 70 micron, 100, 130 micron chips with the associated nozzles, giving you a wide variety of cell types for your applications. You can sort into tubes for enrichment. You can then come back and do your molecular or your sequencing, next gen, or whatever you need in plates. You have that index data. There's default sample cooling. Again, what's the sense of sorting cells if they're going to die? Well, you can chill them and maintain that viability. High throughput is also achieved with the SH800. So again, you can sort at really high rates. So you can sort 12,000 events per second. You can be running, 30,000 events per second in yield mode. So depending on the type of mode you use, you might even be able to get a little higher. But when you combine all these factors, it's easy to see that, even though it's simple assay, having this system in your lab is going to increase your productivity, help you optimize your assay, and you can rest assured the system's easy to use, it's capable, and you're going to have the throughput you need. Moving on to stem cells. Again, stem cell technology and certainly research has really blossomed over, the decade or so. I think we're all exposed to this on bits and pieces in the literature. But, as you may know, a lot of times what happens is there's, multiple sorts that need to occur, for this application. So initially, you isolate targets of interest from a tissue. You then sort those cells and plate them out. You may drive differentiation. You may purify those cells. And then maybe after some workflow, maybe you're going to engineer those cells, right? You're going to get them to become something else. They're going to form an organ or they're going to do something downstream, right? So maybe not all of those cells, have been successfully engineered. So you go through another sort or selection process to enrich and purify those cells. And then you're ready to take those cells to your downstream application, which might be, drug screening. It could be stem cell therapy. Maybe this is in an animal model. But, what I've used this system for is really about, disease modeling, mechanistic studies. You get those isolated, purified, defined cells, and you tease them out of a complex system. And then you can break that system down into its bits and pieces and study, maybe how to modulate a response or mitigate a response. And so being able to isolate cells, purify them once you've engineered them, you can then do your critical steps to get insight into the biology of a specific cell. Again, none of this is complicated, and certainly the Sony SH800 makes it really simple for your lab to do this. The Quantum Lab at Johns Hopkins University uses the SH800. They use it to study cardiac abnormalities, again, targeting stem cells that are derived from cardiomyocytes. As you may know, some cells, are, not the easiest to sort, to work with. And cardiomyocytes are no different. They happen to be, somewhat large, a little rectangular. There's some branching, right? So, sorting these cells requires, an instrument that's capable of doing that. And so you need a larger diameter stream. You need a larger nozzle. You need bigger droplets. You need lower pressure for viability. And so what Peter Anderson shows and what he, is on the record saying is that, the SH800 with the 130 micron chip is able to get his cells not only sorted, but maintain their viability and functionality upon sorting these cells. And so just to kind of show a little bit of this workflow, doing the cardiac progenitor cell isolation and growing it out and having that viability, we have this workflow here. So, you isolate the cardiomyocytes. They're grown in culture. They're tagged with red fluorescent proteins such as TD tomato. The cells are then sorted with the SH800 based on that TD tomato expression. In this case, we kind of target that expression plus viability and functionality. This was determined 2 days after sorting. For experiments where cardiomyocytes were then injected in rats, the cells were observed to transition into adult cardiomyocytes after 1 month. In both cases, the 130 micron chip was used to accommodate these larger cardiomyocytes. And so if you just go through the protocol here, you isolate your cells, you tag them, you sort based on TD tomato, you inject them into the ventricle. Here's your rat recipient. They grow up, they become adults. Here we have our pre-sort and our post-sort. We go through successive rounds of sorting, and here we show the viability after 48 hours. Benefits of the SH800 in stem cells. Again, this is going to repeat itself. The ease of use, automated startup, calibration, and sorting. Again, you don't have to be an expert. The system's going to start itself up while you're not there. You come in, you turn it on, get calibration going. Compensation wizard can help guide you for maybe more complex panels. Again, can run up to 6 parameters. You have some junior grad students. If you have people that come from a non-flow background, compensation is critical, right? You do it wrong, you might not get the data you want. You might not get the cells you want. Compensation wizard guarantees the ideal matrix every time. If you're doing the same assay, you want to maintain consistency between technicians, grad students, postdocs. Again, you can create templates. You can run an experiment based off of templates. So again, this helps with ease of use. Capability, again, the variety of nozzles, sorting into tubes or plates. Sometimes you need to do both throughout the whole process. You initially isolate cells, then you grow and differentiate, and now you want to sequence and you use those plates. Again, sample cooling is a must. Performance, the system gives you that flexibility of maybe an ultra gentle 130 micron chip or speed with a 100 micron chip. The main point, I think, is you have flexibility in how you use the SH800. Moving on to cell biology. So research insight into tumor cell sorting for RNA-seq, single-cell transcriptome profiling of the PDX or patient-derived xenograph. Here's where we take tumor from, let's say, a human, and we're going to isolate cells and inject that into an animal model. And, then we can, study this downstream. Well, why do we want to work this model? Why do we want to sort? Well, intratumor heterogeneity poses challenges to RNA profiling. When you run non-enriched samples, sequencing reads could be noisy, or what's really the read that you want to go with. You have to make a call on something. The purer your sample, the better your reads are going to be. So you need to improve sequence resolution for marker-based treatment calls, to understand maybe expression profiles of the cells of interest. You need these improved sequencing resolutions. Moving into those, tumor cell sorting for RNA-seq. Again, viability of tumor cells is, that's kind of usually the minor fraction of a tumor, right? So, there's the immune infiltrate. There's the tumor itself. There's usually a lot of necrosis. You probably don't want the lymphocytes, right? You might want to sort them out. You're more interested in the tumor. The 10x platform, the Chromium platform, can be used to, profile the transcriptomes associated with disease and help with, clinical outcome, maybe help derive treatment calls and best practices. How could we best, for this particular tumor, these particular markers, get an outcome that's, gives us the best prognosis. That's what we're looking for. Well, you need to have a clean sample in order to get those reads. And this just really highlights, isolating your cells, purifying those cells, removing lymphs, giving you a population that you can then, sort and get your genomics data. The benefits of the SH800 cell biology. Really, it's about, again, ease of use, walkaway operation, processing of clinical samples. Again, compensation wizard, templates. It's going to be consistent. It's going to allow you to come back, at all hours of the night. Because remember, one thing you learn about tumors and translational research, it's hard to predict when they're going to show up. Having a system that's consistent, having a system that you can have an assay and a sort based on a template. Again, having compensation wizards so you're not fiddling, with spillover concerns. Not only gives you the ease of use, it's ease of mind that you're going to do the right thing, because again, these are precious samples. Having the capability of 6 colors, index sorting, certainly having the 130 micron option in terms of a nozzle is critical for tumors. Again, you have to disassociate this tissue. It is going to be, membranes are going to be somewhat compromised, so you need to sort gently. And with a 130 micron chip, you get a low 9 psi, so viability is, unquestionable. You're going to have a great viable yield. And again, performance. You can sort fast if you need to. You can sort slow and safe. There's flexibility. So you're going to get that performance you need for your multitude of workflows, and giving you those critical features and that flexibility. Again, in your lab, when that tumor comes at 2 a.m, and I've been there, the core doesn't open till 8, but that doesn't matter because you have your own SH800 in your lab and it's easy to use. Now we'll move on to genomics. In this example, we'll review how sorting has become a vital step in single-cell sequencing studies. These studies are performed using cells harvested from attacked organs, maybe cell lines, blood, PBMCs, tumor tissue. Cells harvested by chemical, enzymatic, or mechanical methods, for the liberation of single cells for, downstream molecular-based assays, what ends up happening is you have a couple things, right? And none of this is shocking. You have a lot of dead cells. You have a lot of debris. You have a lot of non-viable nuclei. So, that's really what you're looking for, right? And that's all because of, the state of the tissue, the physical, processing, enzymatic processing as well. So the idea here is you need to get rid of unbound DNA and RNA, and thus, have a clean sample, right? So, removing non-viable cells or nuclei, this kind of sorting eliminates those dead cells, those bad nuclei, gives you that, that gives you what you need, your clean, intact nuclei for sequencing. And then mixed populations, right? Because, no tumor is just one population. You have an immune fraction, you have stromal, you have tumor cells. But you're really interested in, just cells of interest, right? Maybe CD8s, maybe it is the stromal. Whatever it is, again, you have 6 parameters to choose from on the SH800. You can use those parameters. You target those parameters, enrich your cells. And again, you make sure they're viable and intact. You get those nuclei, you get the cells you need, and you get clean data. So, there's some old workflows, there's new workflows, there's a lot of ways to go about genomics, right? Some offer you better outcomes than others. So if you don't enrich, if you don't purify your targets and you go to analyze RNA, you may have less than 10% of your sequencing reads are associated with intact nuclei or cells, right? But if you're able to, create your single cell suspension, purify those viable cells, those intact cells, maybe those nuclei that seem to represent, full intact nuclei, you then get reads that are better quality. You can interpret that data with, it actually just streamlines and, increases your productivity because, a few of these experiments are equal to, several of these less quality experiments. Research insight, sorting intact nuclei for epigenomics. Again, you disassociate cells, mechanical or enzymatic approaches. You apply these cells, to certain buffers that are conducive for, your downstream application. It's critical to use DNA dyes, whatever your flavor you want to use, right? Whether it's PI or 7-AAD, you want to identify those intact nuclei. And what this gives you at the end of the day is single, viable, intact, high-quality nuclei for high-quality nucleic acids for those downstream reads. In this last example, just want to kind of go over how the Sony cell sorter is integrated into a part of a workflow. From 10x Genomics, here we have a complex starting sample. It could be PBMCs such as this sample, but it could be almost anything, right? It could be a tumor with an immune infiltrate, could be a lymphoma that you've kind of dissociated. But anyhow, needless to say, you get your cells, you create a single cell suspension. You then apply a variety of total seq antibodies for gene expression profiling, surface marker antibodies to identify and sort those target cells, and a dextramer panel for VDJ profiling. And as always, and what I always recommend to my customers, it's pretty good idea to have a viability marker, right? Because you want to gate on viable cells. You then run this mixture together, you then sort your cells, and you can get, in this example, purified CD8 cells, right? And that dextramer panel in this workflow allows for detection of very low affinity antigenic specific T cells. Once you have your purified cell product, right, so you label your cells, you then sort those cells, you can then move into that multi-omic, study, whether it's gene expression, you want to look at TCR clonotypes, maybe you want to do some phenotype work. Again, there's a variety of things you could do with this purified cell fraction that, in conjunction with, 10x Genomics, the SH800, gives you the power to remove cells that you don't want, get those cell populations of interest, use the power of those 6 parameters, and do your downstream multi-omics. What are the benefits? Again, ease of use. Do you need people who are fully trained? Not at all. They can learn this system in, really just a couple days. You can do bulk sorting. You can do plate sorting. You have those 6 parameters plus your forward side scatter. You can use those parameters to, tease out specific cell types. Again, a template for consistent assay workflow. Capability. You can sort into tubes or plates, right? You have that gentle chip size, the 130. Index sorting. Again, you got some results. Why do these cells, have this certain read and these other cells, have this kind of read or maybe don't perform or, treatment doesn't, seem to show efficacy. Going back to maybe your sorting data and maybe those expression profiles is critical. You have that data with the SH800. Again, performance, I know I'm probably repeating a lot of the same things again here, but, you have what you need to get gentle sorts to get intact nuclei or cells, to get purity, to get throughput, with this system. Again, it's in your lab. You can use it when you want, whether it's Friday night, Saturday morning. It's all about productivity, and having this system in your lab gets you that. Now, just want to summarize. The SH800 cell sorter delivers many features to support cell sorting in many different applications, including automated setup, microfluidics for high throughput, gentle sorting conditions for a wide range of cell sizes. Exchangeable sorting chips, really do prevent, carryover of cells or cross-contamination. The power of index plate sorting, really comes to light, after you have some experiments where you have some really convincing results and maybe some other results that kind of show some different outcomes. You can use that index plate sorting to kind of tease out, why are these cells performing this way? The SH800 integrates many workflows for various applications, whether it's gene editing, stem and immune cell isolation, obviously the genomic workflows, right? I think I gave some great examples, but, this is just the tip of the iceberg. Everyone does something special when it comes to cell sorting, but these really give you the flavor of what the SH800 can do and how it can work for you. We have resources if you're interested in learning more about the SH800 or maybe some of our other instruments. If you want to go to sonybiotechnology.com. We have brochures on instruments. We have application highlights. If you want to look at like the publication record, webinars, maybe even, a 3D tour of the SH800, this can afford you a chance to really see maybe some more detail, learn some more about the system. Of course, you can always reach out to me, at andy.draghi@sony.com. I'm happy to answer questions. Let me know how I can help. Thank you.
All right. And thank you very much for that presentation. We now will go into the live Q&A portion of the event. If you do have any questions you'd like to ask, please put those into the Q&A box and click send. We'll get to as many as we have time for today. So let's start with, let's see here. I want to sort into a 384 well plate. How can I increase the efficiency of single cell deposition so that I can get over 90% of the wells with a single cell?
Excellent question. Thank you for submitting that. The SH800, along with our other sorters, we always recommend when you're doing plate sorting, going to want to position that plate, align that plate, determine that your droplets are going to, land in the center of the well. Save those coordinates, and then you can perform your sorting. You may want to use the angled plate holder. Again, the 7-degree angle is going to improve deposition, into the plate wells. For getting a good single cell deposition, you're looking to avoid debris, you're looking to avoid aggregates, you want single cells. So it's really about sample prep. You're going to want to optimize sample concentration, viability. You're going to want to reduce stickiness. Things like that are going to help you, improve your efficiency, capture your cells, and get those single cell depositions per well. Good question.
All right. Let's go with, how do you ensure the viability of cells or integrity of the nuclei when sorting samples with the SH800?
Yeah, another good question, kind of applicable across a lot of platforms. It's really all about, using a viability dye, or a way to identify nuclei. So, really, one of your first, maybe fluorescent gates, should be one to allow you to identify viable cells and, exclude those dead cells. If you're sorting nuclei, obviously nucleic acid marker, some way to highlight the intense signal, seen in intact nuclei versus fragmented nuclei. They're going to have less DNA, signal. That's going to allow you to, get your viable cells or get your intact nuclei.
All right. Let's go with how do you ensure? Oh, I think I was the one I just asked. How can I improve the yield of sorted cells or nuclei for using them in RNA sequencing reactions downstream?
Yeah, another great question. Really, a lot of these questions really can have a sample preparation component. It all starts with your sample. So, optimize your preparation. If you're doing a dissociation, you're going to want to create those single cells. You're going to want to stabilize nucleic acids. You're going to want to stabilize intact integrity of cell membranes or nuclei. Buffers to maintain single cell suspension. Any released nucleic acids can be harmful. DNA is really sticky. So you might want to use, maybe a DNase in your preparation to cut down on stickiness. Those kind of workflows are kind of something that's, they're well reported on, but again, you're going to have to optimize a workflow that works best for you. Probably a good opportunity to reach out to an FAS, reach out to us here at Sony. We can provide guidance. Every tissue is different. The stickiness, the viability is always variable in tumors. So a little tweaking of protocols and optimization is probably warranted.
Okay. Let's go. While sorting homogenized tissue preps, do the chips get clogged easily? And if so, are they easily removed?
Well, I wouldn't say they get clogged easy, but if you fail to count your cells or strain your samples, you might find that you do get a clog. Again, with every flow cytometry application, you should be straining your cells prior to running on the system. So we may recommend a 35 micron strain prior to running. If you're using larger cells, fibroblasts, neurons, maybe you got to go with a larger micron size strainer. Strain your cells before you do your sort. Count your cells. You want to have consistency in your sorts. You probably want to run at a pretty similar concentration once optimized with each of your experiments. Those are the things that you can do to prevent those clogs. Now, we do have an ability to back flush and remove clogs in the nozzle. It's a simple icon you click on. Certainly, you can wash your probe. There's workflows to deal with this. So it's pretty easy to deal with clogs, which is one of the great features of the Sony cell sorters. And again, something I mentioned earlier, DNase. That can be used to cut down on stickiness, get rid of cell aggregates, clumps, things like that. Those are the things that are probably going to, pose a problem for you. Good question.
All right. What's the recommended EPS for sorting larger cells using the 130 chip? And what is the downside of sorting too fast?
Yeah. So, we have standard, protocols or settings that we tell people. So, the larger chip, slower sorting. So, usually around 3,000 cells per second, maybe 130 micron chip. But, depending on your type of cells and maybe your sample prep, maybe you actually, get to your, sort goal faster if you dilute your sample out. Maybe you run at a slower rate. So it's really about optimization. I tend to tell my customers, you're working with this type of cell. The first few times you run a sort, it's really about optimizing, not only your yield but the viability, your time at the machine. And so that's going to require some pilot projects or pilot experiments where you tweak a few things to really get you the optimal yield or the cleanest yield. And that's really about preparation, cell concentration, your settings. those are things you want to focus on.
All right. Can we use larger chips to sort smaller cells? For example, can we sort lymphocytes with the 130 chip if we only have the 130 chips?
Yeah, no, you certainly can. Some of the downside one is you're used to sorting maybe lymphocytes with a 100-micron chip. You're used to a certain speed. Because of that speed, your cells can be at a certain concentration. At a 130, you're going to be running slower. You don't want to be aborting if you have your cells at a too high concentration and you're getting, maybe multiple cells in a droplet and it's maybe getting aborted. Optimizing your cell concentration. So you can choose to run a smaller cell and a larger chip. You're just going to have to maybe change some of your sample preparation and how you resuspend it.
All right, excellent. We got time for a few more questions here. Do you need to calibrate the position of a 96 well plate every time I sort?
Yeah, so if I was to do one plate a day, I would certainly just confirm that the droplets are landing in the appropriate spot. So it is best practices to confirm the position of those drops prior to sorting. If you run 5 plates a day, I would say just confirm it once, as long as you're using the same plate, the same 96 well plate, the same manufacturer. Once you set it up once and you keep adding plates and continuing to sort, you're good for that day. Remember, you never know if it's a shared instrument, if someone's made some changes, if things aren't, maybe the way you left it. You need to validate that, right? So these cells are precious you're working with. It took time to get there. It's pretty quick to go through and just confirm drops are landing where they need to land on your plate, prior to doing a plate sort. So make sure, you confirm the droplet location for plates.
All right. For multiple sequential sorts, are the cells washed again between each sort, or can you sort straight from the collection tube?
Yeah, great question. Maybe this depends on, what's best for your cells, right? So when you're sorting, your cells are landing in a tube that has some sheath fluid. Hopefully they're landing in a 2x cell culture media or something that's conducive to cell viability. You could run from, that collection tube. So you could do another sort right out of your collection tube. But if what they're landing in is mostly the sheath buffer, if your cells are sensitive to maybe being, not in the ideal media, maybe what's best for you, is to, process those cells a little bit by, adding some more media, maybe straining them if they're sticky. Something you're going to have to, optimized based on your specific needs. But I can tell you, people often will do a second round of sorting right out of the original tube, but that workflow works for them. So just confirm. Good question.
All right, so it looks like we've got time for just this last one here. Can we combine a magnetic bead separation protocol prior to sorting with the SH800 to eliminate some of the non-targets, or would that interfere with the sort?
Yeah, that's a good question. I have customers that certainly do some of this. You want to make sure that your beads are removed from your sample that you're going to be sorting. Again, this is probably something where you want to reach out to your FAS. There's a lot of different bead systems for purity, enriching. So do people do it? Yes. Not every bead's the same. So reach out to your FAS, talk to them about what you're looking to do, the system you're using. Chances are it can work for you, but there are probably examples in which it can't. But hey, talk to us. People do this, but we don't have, it's not a universal answer, right? Because everyone's got a different type of bead. Good that's a good question. People do, use beads to, enrich or clean a sample prior to sorting.
All right. Well, I'd like to thank you guys for your time today and your important research. We'd like to thank LabRoots and our sponsor, Sony Biotechnology, for underwriting today's educational webcast. I'd also like to thank the audience for joining us today and for their questions. This webcast will be able to be viewed on demand. LabRoots will alert you via email when it's available. We do encourage you to share that email with your colleagues who may have missed today's live event. And until next time, thank you very much and goodbye.